Welcome to this tutorial on question 2. Let's get after it. Question A starts with, given the diagram below, state whether this is a DNA or RNA and list a couple clues that led us to this conclusion. So first we're going to see that we have a nice little phosphate sugar backbone over here. And you can see the sugar over here. It's like a cyclopentane with oxygen in there. I don't really know the exact organic chemistry terminology for that, but yeah, we have a alternating phosphate and sugar backbone on this side, and we have one on this side. So you can see the sugar, phosphate group, sugar, phosphate, etc. And so that leads us to two strands, and so that's a huge clue that this is definitely going to be a DNA. But let's look a little further. This is just the basic structure of deoxyribose and ribose, and the biggest difference here is that we have one OH group on this ring, and ribose has two. That's why it's called deoxyribose, because it lacks that other oxygen. So if we go back to our sugar backbone, we notice that in all this, there's only one oxygen there, not two or two OHs. And so another huge thing, and then we have our same thing down here. Huge thing that tells us that this is a DNA molecule. So yeah, two strands, and structurally, from the organic chemistry level, it's just definitely DNA. So now let's go to question 2, or B I should say. What is the sequence of bases in the left strand from 5 to 3 prime? Explain how you determined each base and the direction of the sequence. So for firstly, the direction, we have, I put 5' up here and 3' down there, and let's talk about why. Same thing over here, it's bidirectional, so anti-parallel, 3', 5'. And we can do this by counting our carbons on this ring. And so we have our 1 carbon there, 2 carbon there, 3 carbon there, and that's 3'. That's where it ends. And then if you go down here, we have 1, 2, 3, 4, 5, 5th carbon. It's the end of that strand, so that's going to be 5'. Also, a little tip is that the OH is always connected to the 3'. So whenever you see the OH on top, you know it's going to be 3' up top. Next, what is the sequence of bases? So now we're looking in this middle section here. Yeah, so how are we going to figure that out? We have to look at the structure of the bases, the nitrogenous bases. And I wrote them down over here, and we have some adenine, looks like this, with like a cyclopentane, and cyclohexane with like an amine group, an amide group, or methyl group, that's type of stuff. And then the biggest difference between these two is that you have an amide group there, and you have a carbonyl group there. So they're pretty similar structurally, with the kind of the benzene ring with some other stuff in the cyclopentane there. And they look similar, but this and that are the biggest indicators of adenine and guanine. So let me go over here and to look at cytosine. And this is just your basic, this might be called, I forget the term in organic chem, it was like meta something, amide, whatever, it doesn't really matter. But yeah, you have your ring here, carbonyl group, amide group there. And these are also kind of similar, but here you have that extra double bond and 2 carbonyl groups. And yeah, that's cytosine and thymine. So if you look back over here, definitely we're looking at A's and G's, because we have this cyclopentane and cyclohexane looking deal. But we're on this side, we're not seeing any carbonyl groups, so that tells us it's an A. Let me write this down real quick. The first one's A. Same deal over here, but we have this carbonyl, so it's going to be G. And these look pretty identical, so that's also A. And same deal, G. Alright, and then, so I think that answers question 3. Question 2. So draw an adenine, for 3, we have draw an adenine nucleotide monophosphate attached with a phosphodiester bond. the 3' end of the left strand. Alright, so basically I drew this out over here. We have to draw from our 3' end. This H would leave in solution and it would connect and create this phosphodiester bond here. And then that would give us then to alternate down to our sugar backbone there, so our adenine can attach from there. So it kind of looks like this. I hope you can get a good look at that, what's happening there. And that's what adenine looks like. But essentially it's the exact same thing as up here. Just gotta draw this out. This is the same as that. And then this is over there. Oh, I should probably draw my O right there. That's definitely needed. And actually it might be OH. Okay, so yeah, and then, you know, you can assume that these have H's on them. I'm not going to draw all those out. And then, yeah, so that's how we draw the adenine on there. And then, now it's asking us to draw its complementary nucleotide attached. On the 5' end, yeah, and then fill in the hydrogen bonds. So same idea as earlier. But instead we have start with our phosphate here, so we can just connect our sugar here, draw our phosphate, get the 5' end going, and then we kind of just copy up here, because like we already have our adenine drew right here. So what do we have up here? A little thymine action. So we can draw that in. That's what it looks like, as I showed you earlier, and that connects from there, just like how it connects from there up there. And then our hydrogen bonds are right in here. You kind of drew them there. As you can see, there's no hydrogen bonding up here. These are a little too far away to hydrogen bond, but yeah, that's how you draw that out. I think that answers the first 4 questions. So let's dive into our what if question. So I think what I want to do here for what if, let's say that this is our template strand for a transcript, or the process of transcription. So let's write this template out. Sorry, I got some notes over there. A little messy, but I think you will be able to see what's going on. So TC. Alright, so that's our template strand, and so mRNA, so let me erase this C. There's a lot of C's and G's and T's and A's all over the place. Alright, so our mRNA is going to add 5' to 3' along this template strand. And funny thing about RNA is that instead of A, when it sees a T, it gives it a U. And C, classic, G, another U, and another G. So that's the direction it'll run. It's going to be adding along here as its base pairing, RNA polymerase. Yeah, so it's just a short little transcript, but that's how that would work if this or this was our template strand in transcription. Alright, I think that answers everything. Cheers.